Okay, so welcome back to this next video in which we are discussing pain and analgesic drugs. Okay, right, so we have now looked in detail at how you get a pain response to this simple case where you deliver a stimulus that is capable of causing tissue damage, okay, and how that produces pain. Okay, what we now want to talk about is how, if you actually do have tissue damage, potentially because of an injury or potentially because of infection, how does that cause the prolonged pain that we all know it does cause, okay? And this type of pain is known as inflammatory pain because sites of injury and infection are sites of inflammation. Inflammation occurs at sites of injury and infection, and it's many of the mediators of inflammation that cause the pain that's associated with inflammation. Okay, so we're now going to look at inflammatory pain, which is in tissues that have been damaged either by infection or injury, so following either an injury or an infection. Okay, right, so firstly let's just talk about inflammation itself. So I'll start off by telling you the cardinal signs of inflammation. Okay, so there are four main cardinal signs of inflammation, and they're stated in Latin usually, just for pretentiousness. Okay, so the first is rubor. So a site of infection becomes red is what this means. So redness is what rubor means. Okay, then you also get tumor, swelling. Okay, so this again is the Latin for swelling. Okay, then calor, which means that it's going to become a hot area. And then the final one is what's known as dolor, which is pain, basically, and the inflamed area becomes painful. And we want to explain why the inflamed area becomes painful. That's our aim. Okay, so firstly let's just do a brief overview of what occurs in inflammation to cause the other cardinal signs of inflammation, so tumor, rubor and calor here, and then we'll look in detail at how you cause this fourth one, dolor, the pain. Okay, right, so basically in a site of inflammation what happens is the blood vessels leading to that site dilatate. Okay, so the arterioles, the small little blood vessels carrying arterial blood that lead on to the capillaries, those are going to dilatate. Okay, so let me just draw the microvasculature here. So firstly you have the little arterioles, so here is an arteriole. Okay, and then what's going to happen is the arteriole is going to split into the microvasculature even more. So it's going to split down into tiny little blood vessels known as capillaries, and then those capillaries will reconverge into venules. Okay, right, so this is capillary bed here, and this is a venule here. Okay, so those are the different parts of the microvasculature, and I'll just colour them in differently. So here are the capillaries in turquoise here. Okay, here is the arteriole in red here. Okay, and then we've got the venule here in blue. Okay, so one of the key things that happens in inflammation is that the arterioles leading to the tissue that has been damaged are going to dilatate. Okay, so the arterioles have smooth muscle cells surrounding them, which the capillaries and the venules don't. Okay, and these smooth muscle cells can control how wide the arteriole is, and therefore how much blood is going to flow through here. Okay, so these arterioles are going to dilatate. Okay, or just dilate, whichever terminology you prefer. Okay, they both mean the same thing. Okay, and this is going to allow more blood to flow to this affected area. Okay, now that's what's going to cause the rubor, the redness, because now more blood is going to that area. And it's also going to cause the calor, okay, because blood is hot. Okay, and that's going to make the inflamed area hot. Okay, so rubor and calor are caused by the increased blood that's going to go to an area because of the dilatation of the local arterioles which lead to that area. Okay, then the other major thing that happens is within the capillaries and the venules. Okay, so the other major thing which occurs is changes to the endothelial cells which cause them to contract. So you get a phenomenon known as endothelial cell contraction. So my abbreviation for endothelial cell here is EC. So this stands for endothelial cell. So the endothelial cells are those little cells that line up blood vessels, and in the case of capillaries and venules, the walls of capillaries and venules pretty much just consists of endothelial cells sitting on a basement membrane. So let me draw this. Okay, so here is an endothelial cell here, okay, and I'll have it sitting on a basement membrane. So the wall of a capillary, or the venules that just follow the capillaries, they literally just consist of endothelial cells sitting on a basement membrane. So this is the lumen of the blood vessel, okay, and this is the extracellular fluid here. Now usually the endothelial cells are nice and close together, okay, and they have junctions between one another which stop too much fluid from leaving the blood and going into the extracellular fluid. Especially what happens is they stop proteins from the blood generally leaving the blood. Okay, so usually this means that proteins have to stay in the blood. Okay, so little molecules like oxygen and carbon dioxide and water can exchange, but big molecules like proteins can't get across these walls. Okay, now what's going to happen in inflamed areas is these endothelial cells are going to contract. Now let me show you what's going to happen. They literally are going to contract. They're going to withdraw their peripheral aspects, and it's going to now start looking like this. Okay, so they're going to open up big gaps between the endothelial cells, and it probably is a little bit over-exaggerated what I've shown you here, but this gets the point across. And the endothelial cells are going to contract, and this is going to open up new spaces in the walls of the capillaries and the venules, and now what can happen is fluid and proteins can move out of these blood vessels and into the extracellular fluid. And I want to stress that this is going to happen in not just the capillaries, but also the venules just after the capillaries, and there's a special name for those tiny venules that are just after capillaries. They're called post-capillary venules. Okay, so you get endothelial cell contraction in the capillaries and the post-capillary venules. Okay, that's going to allow fluid to leave the blood lumen, the blood vessel lumen, and go into the extracellular fluid. Okay, and it's going to bring many proteins that would not usually be able to get out of the blood into that extracellular fluid. Okay, and this new fluid that you form is what's known as inflammatory exudate, or just exudate. Okay, so you're going to form a large amount of exudate basically at the inflamed area, and this is one of the key things that causes the swelling of that area, the tumor. Okay, in addition, the endothelial cells also do one other thing, which is they start to move whole cells out of the blood vessel lumen and into the extracellular fluid. Okay, so they start moving white blood cells out of the blood and into the extracellular fluid. Okay, so white blood cells are involved in fighting infection and also involved in repairing damaged areas of tissue. Okay, now the fancy word for white blood cells is leukocytes, and that covers all the many different types of white blood cells. Okay, and basically when an area is inflamed, the endothelial cells are now going to acquire the ability to help white blood cells, such as the one that I'm showing here, to move out of the blood vessel. So although the gap I have shown here is absolutely enormous and it looks as though it would just be able to get out on its own, basically it's not quite as extreme as this, and instead to get out of the blood vessel, a white blood cell actually needs help from the endothelial cells to allow it to squeeze through this gap between the endothelial cells and into the extracellular space. Okay, so this phenomenon of leukocytes leaving the blood vessel is known as leukocyte extravasation. Okay, extravasation means leaving the vascular system, going beyond the vascular system. So leukocyte extravasation there. Okay, so those are the three key things of inflammation. Vasodilatation, which causes rubor and calor, and then endothelial cell contraction, which produces the exudate, and endothelial cell leukocyte extravasation, which recruits leukocytes from the blood into the site of inflammation, and that also contributes to the swelling, because these cells are big things. Okay, right. So, those are the key parts of inflammation then. Okay, now, the other thing to say about an inflamed site is that you get absolutely loads of molecules floating around in the extracellular fluid that would not normally be there. So in an inflamed portion of tissue, so let me just draw a square so that I've got something to point at. So let's say this is our inflamed area of tissue, so loads and loads of cells are in here, and this tissue is undergoing inflammation. Basically, floating around in the extracellular fluid, you get loads of molecules that would not usually be there, which contribute to the maintenance of the inflammatory state and also contribute to the pain. Okay, and we're going to see several examples of these in a moment. Okay, but collectively this is known as the inflammatory milieu. Okay, this collection of all of these new molecules in the extracellular fluid that are pro-inflammatory and often pro-pain, that are present in the extracellular fluid at high concentrations which would not usually be there. The collection of all of them is this umbrella term, inflammatory milieu, covers all of them basically. So there is this the presence of this inflammatory milieu in these sites of inflammation basically. Okay, so now let's turn our attention to this final cardinal sign of inflammation, which is dolor. How do you actually get pain at a site of inflammation? Well, there are two parts to this, okay? One of the parts is that the polymodal fibers, okay, the polymodal fibers start firing tonically, basically. Okay, so the polymodal nociceptors start firing action potentials tonically. So where should I put this? So they start setting up action potentials tonically. They're just firing action potentials off again and again and again. So if I draw one of these polymodal nociceptors here. So here is the polymodal nociceptor here, and then of course it will be attached to some C fibre here. Okay? And this C fibre will then go off and enter the spinal cord in the way that we've previously described. Okay, now, so one of the key things that causes inflammatory pain is that these polymodal nociceptors are going to start firing action potentials tonically because of the presence of the inflammatory milieu, and we will look at explanations for that in a moment. Okay, there are many different explanations for that. Firstly, many of these molecules directly stimulate the polymodal nociceptors, but also prostaglandins are capable of giving these polymodal nociceptors a tonic firing rate on their own, similar in fact to the way that the heart has a tonic firing rate. The cardiomyocytes will tonically fire, okay, and we'll come back to that in a moment. It's a very interesting part of the action of prostaglandins. Okay, right, so that's one of the parts of the inflammatory pain. The other part is what's known as sensitization of all nociceptors, so sensitization of all nociceptors, so not just the polymodal nociceptors, but also the thermal nociceptors and the mechanical nociceptors, which remember are attached to A delta fibers rather than C fibers. Okay, so you get sensitization of nociceptors. Now, what does sensitization mean? Well, sensitization can be split into two separate phenomena. Okay, so let me put these here. So the two separate phenomena of sensitization are allodynia, okay, and also hyperalgesia. So let me explain what allodynia and hyperalgesia are and how they differ from one another. Okay, so allodynia firstly. Allodynia means that you get pain in response to stimuli that were previously innocuous, i.e. stimuli that previously would not have caused pain are now going to cause pain. Okay, now we're all familiar with a nice example of allodynia. Okay, so let's say you've been sunburnt. Okay, you've been lying on a beach and you've got sunburnt. okay? If you then go into a hot shower, it will really, really hurt. Okay, now previously, before the sunburn, going into a hot shower would not hurt. Okay, but now, once you've got that inflammation occurring all over your skin, when you go and expose yourself to that hot water, okay, which was previously an innocuous stimulus, it now hurts. So allodynia is all about previously innocuous stimuli now causing pain. Okay, so in stimuli that previously didn't cause pain, which I'll call innocuous stimuli, now cause pain. And the example that you should always think back to is the example of sunburn and showering. Okay, now, how is hyperalgesia different from that? Hyperalgesia refers to the fact that if you have stimuli that did previously cause pain, okay, so potentially thermal stimuli or mechanical stimuli that were previously pain-causing, so they were powerful enough to activate nociceptors. Well, now what's going to happen is the pain that you're going to experience for those same stimuli is going to be bigger, okay? So noxious stimuli that previously did cause pain, they now cause much more pain. Okay, so hyperalgesia, too much pain. Algesia is all about pain. So literally too much pain, basically. Okay, so really what sensitization of nociceptors is all about doing is it means that these nociceptors are easier to activate, basically. Innocuous stimuli, less intense stimuli, can now activate them, and the previous stimuli that did activate them now produce more firing, basically, than they would have previously. The rate at which the action potentials are going to be discharged is going to be greater. And I probably should have said that at some point. So intensity of stimulus within these nociceptors, as always, or as is common in neurobiology, because the action potential is an on-off signal, you have to code intensity of a painful stimulus by the frequency at which action potentials are firing. Okay, so basically the phenomenon of hyperalgesia is that a noxious stimulus that was previously pain-inducing is now going to result in a faster frequency of action potential firing, okay, which will therefore result in more pain. Okay, so noxious stimuli now produce more pain. Okay, so that's the part of sensitization of nociceptors, okay, and this is going to occur for all of them, okay. Basically, all of the different nociceptors, the thermal nociceptors attached to A delta fibers, the mechanonociceptors attached to A delta fibers, and the polymodal nociceptors attached to C fibers, all of them are going to become much easier to activate, okay, and this is going to mean that if you do expose this area that is currently inflamed to noxious stimuli, let's say you deliver a noxious mechanical stimulus, it's going to hurt much more than it would have previously. Okay, so that will be called mechanical hyperalgesia. In addition, if you deliver a noxious heat stimulus to the inflamed area, you're also going to get much more pain than you would usually get, and that will be called thermal hyperalgesia. So there are all sorts of different components to this, and of course there's mechanical allodynia and thermal allodynia as well. So those are the different components then of sensitization, and we'll come on to the mechanisms which underlie this in just a moment. Okay, right, so what we now want to see is how the components of this inflammatory milieu cause these two things. How do they cause the polymodal nociceptors to be firing continuously, and how they cause this sensitization of nociceptors? So one point I should probably stress is that the A delta attached nociceptors, the thermal nociceptors and the mechanical nociceptors, those are not firing tonically, unlike the polymodal nociceptors. These ones fire all the time whilst you've got the area inflamed. Okay, but the A delta attached ones don't. Okay, they're still only going to fire if you actually deliver some stimulus, some thermal stimulus or some mechanical stimulus. But the key thing to understand is that you've now got sensitization of those. So that means that you've got allodynia, where previously innocuous thermal and mechanical stimuli are now going to cause pain. Okay, they're going to cause the activation of those A delta attached nociceptors, and also you've got hyperalgesia, whereby previously noxious stimuli, if applied to this inflamed area, are going to activate A delta attached nociceptors even more, and therefore cause a more intense signal to go to the brain and therefore cause more pain. Okay, so you've got tonic activation of the polymodal nociceptors, but not of the thermal and mechanical nociceptors. But you do have sensitization of those, such that if a thermal or mechanical insult is made, the response is going to be exaggerated, basically. Okay, so we now want to look at the underlying mechanisms for this. We want to look at how the inflammatory milieu actually causes this. So we're going to start off by looking at certain things which can activate polymodal nociceptors directly. Okay, we're then going to turn our attention onto the prostaglandins and all the incredible things they do, and that will allow us to discuss the mechanism of action of the non-steroidal anti-inflammatory drugs, the NSAIDs. Okay, and then we'll move on to bradykinin and its role in all of this. Okay, so firstly, things that are going to activate polymodal nociceptors directly. Well, some of these we've talked about earlier. So when you have tissue damage, you're going to initially get ATP being released into the extracellular fluid. Okay, and this will initially form part of the inflammatory milieu. Okay, and this high level of ATP is then of course going to activate those P2X3 receptors, which remember were homotrimers of P2X3 subunits. I'll try and just find the picture for this. Okay, have I got it? Here we go. So here is our trimer, and the P2X3 receptor was made up of 3 separate P2X3 subunits. Okay, and remember it responded to ATP. It was an ATP-gated ion channel. Okay, so that then is going to activate these polymodal nociceptors and cause them to fire action potential. So that's one of the things that is partially going to underlie the tonic firing of the polymodal nociceptors. Okay, another thing is the fact that in this inflammatory milieu, you generally have a low pH. Okay, so a low pH is one of the phenomena observed in inflammatory milieu. Okay, so it's an observed phenomenon at sites of inflammation that you have a low pH. Okay, so high proton concentration therefore. So proton concentration is high, and we know of course that high proton concentration can then activate receptors on the surface of the polymodal C fibers known as acid-sensing ion channels, ASICs, and again this can underlie the tonic firing of these polymodal nociceptors. Okay, so remember these are both chemical stimuli, and those won't activate the A delta attached nociceptors, which don't have the receptors for chemical stimuli. They're thermal and mechanical nociceptors, remember. Okay, right. Something else that I'd like to mention is that generally in inflamed areas you get the production of an endocannabinoid known as anandamide. Okay, so let me just mention anandamide. Okay, so anandamide is an endocannabinoid. Okay, now endocannabinoids have very small roles in the body. Usually they're synthesized at times of problems, basically. They become involved really far more in pathophysiology than they are in physiology. Okay, and indeed anandamide is produced by cells in inflamed tissues, basically. Okay, right, now anandamide is often just abbreviated as AEA for short, and you might be wondering why on earth is it abbreviated to AEA? Surely just AA for anandamide. But it's abbreviated that because of its real name. The real name for anandamide is N-arachidinyl ethanolamine. Okay, so AEA stands for arachidinyl, that's the first A, and then the EA is for ethanolamine. Okay, and this is just arachidonic acid linked to an ethanolamine molecule. Okay, right. So, how can anandamide be involved in tonically activating these polymodal nociceptors? Well, it's believed that anandamide activates the TRPV1 receptor in much the same way as capsaicin would activate the TRPV1 receptor. Okay, so remember the TRPV1 receptor was one of these noxious heat receptors. Okay, so I'm now just going to draw it simpler as these two portions here, like so. So TRPV1 ion channel, remember, was a tetramer of 4 TRPV1 subunits, okay, and when it's opened, it allowed sodium and calcium ions into the cell, which caused that receptor potential. Okay, so anandamide is capable of binding to TRPV1 receptors and causing them to open. And again, this could underlie the tonic activation of the polymodal nociceptors. The little difficulty there that arises in trying to explain this is why wouldn't that also cause the activation of the thermal nociceptors that are attached to A delta fibers? Okay, well, remember that in order to actually get the primary nociceptive afferent to fire an action potential, you usually need more than one. You need a lot of receptor potentials to add together to actually cause an action potential to be generated. So potentially anandamide is just contributing with loads of other things to build up a big enough sea of receptor potentials that the primary afferent nociceptor fiber actually fires an action potential. So maybe it isn't a big enough stimulus for the A delta fiber attached thermal nociceptors to actually fire action potentials tonically in response to the presence of the anandamide. Okay. So maybe this contributes with these things to produce the tonic activation of the polymodal nociceptors. Okay, so those are some things that can then directly activate some of the receptors that we've seen. Okay, now what I want to turn my attention onto is the prostaglandins, okay, which are incredibly important, not least because their production is the target for a very widely used branch of analgesic drugs and anti-inflammatory drugs as well. Okay, right, so let's talk about them prostaglandins. So prostaglandins are actually going to be involved in both of these phenomena that I have outlined as being the cause of the problems in inflammatory pain. Okay, they're going to cause polymodal nociceptors to fire tonically, and they're also going to cause the sensitization of all different nociceptors. So what we're firstly going to look at is we're going to look at the mechanism by which they sensitize all nociceptors, by which they make nociceptors more likely to fire action potentials to innocuous stimuli and fire more action potentials to previously noxious stimuli. Okay, and then what we'll come back onto is potentially how in C fiber attached nociceptors, these polymodal nociceptors, potentially it's going to set up this rhythmic pacemaker activity. Okay, right. So prostaglandins then firstly. The first thing to say is that prostaglandins are produced by all cells. All cells have the potential to produce prostaglandins. Okay, so all of the cells in this inflamed tissue can start producing prostaglandins. Okay, now, let me say how you actually produce prostaglandins then. So the first thing to say is that prostaglandins are produced from the precursor arachidonic acid. Okay, so I'll abbreviate arachidonic acid down to AA, but I think I should probably write it out in full at least once. So this is arachidonic acid. Okay, now we're not going to draw the structure of arachidonic acid, we'll just draw a nice flow diagram for this. Okay, so arachidonic acid is going to be converted firstly into prostaglandin G2. Okay, so in the synthesis of prostaglandins, these are the first reactions that occur. First, it's converted into prostaglandin G2, and then it's converted into prostaglandin H2. And both of these reactions are catalyzed by cyclooxygenase enzymes. Now, in inflamed tissues, there is a certain form of cyclooxygenase that is upregulated hugely, which is called cyclooxygenase 2, or COX-2 for short. So there are two main forms of cyclooxygenase, cyclooxygenase 1 and cyclooxygenase 2. Cyclooxygenase 1 is expressed in many tissues of the body, and it's endogenously expressed. Whoops, I've written cyclooxygenase 1 there. Cyclooxygenase 2 is majorly upregulated in tissues which are undergoing inflammation. So in this inflamed tissue, the cells will upregulate hugely cyclooxygenase 2. They'll go from not expressing any cyclooxygenase 2 at all to suddenly expressing loads of it. Okay? And the cyclooxygenase 2 can catalyze these two reactions. The first reaction is the conversion of arachidonic acid into PGG2, which stands for prostaglandin prostaglandin, that's the PG, and then G2, and then the second reaction will catalyse prostaglandin G2's conversion into prostaglandin H2, and it's the same enzyme which catalyses both reactions. Now these two reactions are so important that they've actually been given their own names. Okay, so the first reaction, which converts arachidonic acid into prostaglandin G2, that's known as the cyclooxygenase reaction. Okay, and then the second reaction, which converts prostaglandin G2 into prostaglandin H2, that's then known as the peroxidase reaction. Okay, so this will be happening in the cells at the site of inflammation. Okay, and that's the start of the reaction. Now what we can do is we can use prostaglandin H2 to make all sorts of different prostaglandins. So to give some examples, a very important one is that we're going to start producing prostaglandin I2, and another one we can produce is prostaglandin E2. Okay, so prostaglandin I2 has another name. It's known as prostacyclin, and it is majorly involved in maintaining inflammation. It's not particularly involved in pain, but it's majorly involved in maintaining this constantly inflamed state. Okay, so it acts on smooth muscle cells surrounding arterioles and is going to cause continued dilatation, basically, or relaxation rather, which then leads to the dilatation of the blood vessels, which is a major part of maintaining inflammation. Okay, but the one that's of interest for us, studying the pain component of inflammation, is prostaglandin E2 here. Okay, this is major in causing pain. Okay, so let's now have a look at what the mechanisms are by which prostaglandin E2 causes both the sensitization of nociceptors and that tonic firing in C fibers, how it contributes to both of these things. And it does contribute majorly, hence the efficacy of the non-steroidal anti-inflammatory drugs, which block the production of prostaglandin E2 by blocking cyclooxygenase 2. And I'll come back to the non-steroidal anti-inflammatory drugs and give examples once we've looked at the mechanisms. Okay, right. So, how does prostaglandin E2 work on nerves then? Okay, well now it's time for a bit of intracellular signalling. Whippy! Okay, so, prostaglandin E2 then, the nociceptors of all different types, so of the C fibers, of the polymodal nociceptors, the thermal nociceptors, and the mechanical nociceptors, they all have receptors for prostaglandin E2 on their surface. And these receptors are G-protein coupled receptors, so they have this typical 7-membrane spanning alpha helix structure, okay, which all G-protein coupled receptors have. Okay, and the specific prostaglandin E2 receptor that is thought to be extremely important in everything to do with inflammatory pain is the EP4 receptor, standing for prostaglandin, and then for some reason you put the E first, E, and then it's the fourth receptor. So there are 4 different receptors for prostaglandin E2, EP1, EP2, EP3, and EP4. The fourth one is believed to be the one that is really, really important in mediating the pain-producing actions of prostaglandin E2. Okay, so here is our G-protein coupled receptor. Okay, so let's just discuss what the G-protein coupled receptor is then going to act on. Well, it's of course going to act on a heterotrimeric G-protein. Okay, now the EP4 receptor is going to specifically act on GS heterotrimeric G proteins. So let me just talk you through heterotrimeric G proteins. So we'll have a continuation of the plasma membrane here. So heterotrimeric G proteins consist of 3 separate subunits, as their name suggests. Trimeric means that they're going to be a 3-membered structure. Heterotrimeric means that the 3 constituent members of the trimer are all different. Okay, so you've got 3 separate subunits here. An alpha subunit shown here, a beta subunit shown here, and a gamma subunit shown here. Okay, now let's colour those all in different colours. So we'll have the alpha subunit here in vivid purple, okay, like so. Then we'll have the beta subunit here in, no, I won't have it in green, I'll have the gamma subunit in green, G for gamma, Okay, and then we'll have the beta subunit in blue here. Okay, right. Now, prior to the activation of the EP4 receptor by prostaglandin E2, the heterotrimeric G protein is in the off state, okay? And that's the key thing about all G proteins. They have two states. They can be in an on state or an off state. Now, in the off state, the alpha subunit of the heterotrimeric G protein is going to have guanosine diphosphate bound to it. And in the on state, the alpha subunit of the heterotrimeric G protein is going to have GTP bound to it. Now, only when it's in the off state will it associate with these two other subunits, the beta and the gamma subunit. Okay, so at the moment it's bound with the beta and the gamma subunit, so it must be in the off state and it must have guanosine diphosphate bound in that little site, if you can see that. Okay, so that's supposed to be guanosine diphosphate. Okay, right. The other thing to say then is that the beta and the gamma subunits, they never ever split apart. They always remain bound together, and therefore often people will just refer to them as the beta-gamma complex. Okay, so even if the alpha subunit goes into the on state and breaks apart from them, they remain bound to one another as the beta-gamma complex. The final thing to say before we actually go to what happens when the EP4 receptor gets activated is that the heterotrimeric G protein is not an integral membrane protein, but it is attached to the underside of the phospholipid bilayer. Okay, and it's attached by having lipid moieties attached onto it. So the alpha subunit gets lipid moieties attached onto it. And the basic idea here is you attach on a very hydrophobic molecule onto the side of the protein that sticks out off the side of the protein. Okay, now this is hydrophobic, so it's not going to interact with water molecules at all well. Okay, so what ends up happening is it gets implanted into the inner leaflet of the phospholipid bilayer, and that then holds the alpha subunit attached to the underside of the phospholipid bilayer. Similarly, the gamma subunit also has a lipid moiety attached onto it, which I'll just represent as this line here. Okay, so this is a lipid moiety. It's different lipid moieties for the alpha subunit compared to the gamma subunit, but the basic principle is the same. Okay, that it's implanting into the inner leaflet of the phospholipid bilayer and thereby holding this protein that has the lipid moiety attached on at the cytoplasmic face of the phospholipid bilayer. Okay, right. So now what's going to happen is our prostaglandin E... Ooh, I've missed off one key bit here. Okay, in fact, actually, I'll do that in a moment. So, prostaglandin E2 is going to bind to and activate the EP4 receptor. Okay, the EP4 receptor is then going to be capable of now activating certain heterotrimeric G proteins. Now, this is key. G protein coupled receptors do not activate all heterotrimeric G proteins. There are absolutely loads of the things, okay, because there are loads of different alpha subunits for heterotrimeric G proteins, and there are also loads of different beta and loads of different gamma subunits for heterotrimeric G proteins. Okay, now, as far as the signaling properties of the heterotrimeric G protein are concerned, and as far as whether they're going to be activated by a certain G protein coupled receptor is concerned, it does not matter what the beta subunit is and what the gamma subunit is, which is why no one ever bothers to tell you which beta and which gamma subunit is there. It really does not matter. Okay, that's why people don't make a big thing of that. Okay, however, what is really important is which alpha subunit you have here. That really does affect what its signaling properties are and also which G protein coupled receptors can activate it. Okay, now EP4 receptors are specifically going to activate heterotrimeric G proteins which have alpha S subunits. So a specific G protein alpha subunit is the alpha S subunit, and it is heterotrimeric G proteins with this alpha S subunit that the EP4 receptor is going to activate. Okay, now, because the beta and the gamma subunit don't matter, we name heterotrimeric G proteins according to what their alpha subunit is. So this would be called a GS heterotrimeric G protein. Okay, right. So when prostaglandin E2 binds to the EP4 receptor, it's going to cause conformational changes in that EP4 receptor, which is then going to allow the EP4 receptor to bind to a GS heterotrimeric G protein that's in the off state, and it's going to be able to activate that GS heterotrimeric G protein. So it's going to rip that GDP off, okay, and then what's going to happen is a GTP molecule will come in from the cytoplasm and replace that GDP molecule. Okay, so you're going to replace the GDP with GTP. That will then change the conformation of the alpha S subunit, okay, and it will now go into the on state, and in the on state it's no longer going to associate with that beta gamma complex, so they're going to cleave apart. Now both of these two complexes, both of these two molecules, the beta gamma complex and the alpha S subunit with GTP bound, are going to remain attached to the cytoplasmic face of the phospholipid bilayer by virtue of their lipid moieties. So here's the beta gamma complex, and here is our alpha S subunit here, okay, with the GTP now bound to it, which is keeping it in the on state. Okay, so let's colour these in. So here is the alpha S subunit, now coloured in vivid purple, and here is the beta and the gamma subunit. Okay, so now what we want to see is what is the alpha S GTP subunit going to do? We don't care about the signalling properties of the beta gamma complex. They do have signalling properties, but for our purposes I don't care, okay? So we're going to abandon that one, okay? We're going to concentrate completely on what is the alpha S GTP complex now going to do. Well, it's going to go and activate adenylyl cyclases, famously so, okay? So let me now draw an adenylyl cyclase enzyme, and I think I'll just continue the membrane on like so. Okay, so adenylyl cyclase enzymes, the next key player in this intracellular signaling cascade. Okay, so let me show you the structure of an adenylyl cyclase enzyme. It is a single polypeptide, okay, so that simplifies things beautifully. It's not a huge, great multi-protein complex, it's just a single polypeptide, okay, and it has its amino terminus on the intracellular aspect, and then it has two clusters of 6 membrane-spanning alpha helices. So here is this first cluster of 6 membrane-spanning alpha helices. Then we have an intracellular loop followed by a second cluster of 6 membrane-spanning alpha helices, and then the cytoplasmic tail at the C-terminal end is over here. Okay, so let's highlight up some key features here. So we have these two clusters of 6 membrane-spanning alpha helices here and here. Okay, now these have special names. This first cluster of 6 membrane-spanning alpha helices, this is known as the transmembrane domain 1. So this is TMD1. And this second cluster of 6 membrane-spanning alpha helices, this is known as the transmembrane domain 2. So TMD here stands for transmembrane, that's the TM, okay, and then domain. Okay, right. So those two transmembrane domains hold the adenylyl cyclase at the plasma membrane, but they are not, despite looking mightily impressive, they are not the actual active site of the enzyme. Instead, to find the active site of the enzyme, we're going to have to look at these two domains here. So this loop between the transmembrane domain 1 and the transmembrane domain 2, this is known as the C1 domain, and it's split up into two separate portions. The C1A domain, which is the portion closer to TMD1, okay, and the C1B domain, which is the domain closer to TMD2. Okay, and I'll colour these in special colours. I'll just try and straighten this up a bit. Okay, so here is C1A, now in turquoise, and what colour should we have? C1B, and we'll have it in blue. Okay, and here is C1B, now in blue. Now, this cytoplasmic domain after TMD2, this is known as the C2 domain, okay, and it can be split into two separate portions as well, the C2A domain here, and the C2B domain here. And I'll colour the C2A domain again in turquoise, and I'll colour in the C2B domain in an orange. Okay, right, so where is the active site of the enzyme? Well, at present, the active site is not constructed, not the way that I've shown it. The way that I have shown it is an inactive adenylyl cyclase. So this is an adenylyl cyclase that is not doing anything. Okay? To build an adenylyl cyclase that is capable of converting ATP into cyclic AMP and pyrophosphate, what you need to do is dimerize the C1a domain with the C2a domain. When they dimerize together, that makes the active site for this enzyme, the domain which is actually capable of catalyzing the reaction. Okay, so how then does the alpha-S GTP subunit activate adenylyl cyclases? Well, this is what it does. It comes over to the adenylyl cyclase and it binds to the C1 domain, a portion of the C1 domain. It then also binds to a portion of the C2 domain and it brings them very close together. So these two portions end up close together and this promotes the dimerization of C1a with C2a, and therefore it promotes the assembly of the actual active domain, which can then convert ATP into cyclic AMP and pyrophosphate. So the alpha-S GTP subunit activates the adenylyl cyclase enzyme, and I put this the wrong way around. I was about to write cyclic AMP as the reactant. Okay, so the reactant is ATP, and the two products here are cyclic AMP, and then also a pyrophosphate molecule, which are abbreviated as PPI, which is just two phosphate groups attached to one another. Okay, right. So now what has happened is this has resulted in the production of cyclic AMP. Now, for the actual activation of tonic firing within polymodal nociceptors, we don't actually need to go to the next step of this classical signaling pathway. Okay, the cyclic AMP does that alone, and we'll come back to that in a moment. Okay, but I firstly want to handle the hyperalgesia and the allodynia, the sensitization of all nociceptor fibers, before I go on to that. Okay, and that does require us to go to the next step of this classical GS cascade here. Okay, so protein kinase A then we need to go to next. So basically protein kinase A is going to be activated by the binding of cyclic AMP molecules to it. Okay, now protein kinase A unfortunately is not, at least not in the inactive form, it is not a single protein. Okay, it is a multi-protein complex. So let me show you the structure of protein kinase A. Okay, which has another name which I should just mention. It's also known as the cyclic AMP dependent protein kinase. And if you read the literature, they prefer to call it the cyclic AMP dependent protein kinase than protein kinase A. Okay, but protein kinase A is usually preferred among students. Okay, so cyclic AMP dependent protein kinase, just another name for it. Okay, right, so let me show you the structure of an inactive protein kinase A at the moment. Okay, so inactive protein kinase A actually consists of 4 separate proteins all bound together. So let me show this here. So here are 2 of these proteins being shown now. Okay, and now I'll put in the final 2 here, like so. Right, so here are our 4 separate proteins. So these 2 here, which are in these sort of L shapes, like so, with these 2 little invaginations here, which I'll let the cat out of the bag, are the cyclic AMP binding sites. These 2 are known as the regulatory subunits of protein kinase A. Okay, so these are called regulatory subunits of protein kinase A. Okay. So I'll colour these regulatory subunits of protein kinase A in red now. Okay, and there are actually 2 different proteins that are both regulatory subunits of protein kinase A. So there are type 1 regulatory subunits of protein kinase A, and then there are type 2 regulatory subunits of protein kinase A. So let me just talk about this, okay, because this will actually become important in just a moment. Okay, so you can either build a dimer of 2 regulatory subunits that are built out of type 1 regulatory subunits of protein kinase A, which are abbreviated as R1 for regulatory subunit of protein kinase A type 1, or you can build the dimer of the 2 regulatory subunits out of 2 type 2 regulatory subunits of protein kinase A, abbreviated R2. So, when you're building this protein kinase A, you firstly construct the dimer of the two regulatory subunits, and then you attach these other two proteins, which we'll come on to in a moment, onto the dimer. Okay, and when you produce the dimer, you can either make the dimer out of two type 1 regulatory subunits of protein kinase A, or you can make the dimer out of two type 2 regulatory subunits of protein kinase A. And I would not be mentioning this unless it was going to become important later on, and it will do. Okay, so that's why I'm mentioning that now. Okay, right. These other subunits here, which I'll put a C inside, these are the actual catalytic subunits of protein kinase A. So these are the actual ones that are capable of phosphorylating proteins. The regulatory subunits aren't. Okay, so these are the catalytic subunits of protein kinase A. Okay, right. So basically, the catalytic subunits of protein kinase A are capable of phosphorylating proteins. However, when they're bound in this great big tetrameric structure here, they are absolutely not capable of phosphorylating proteins. Currently, they are inactive when they're in this complex. Okay, they're inactivated by the regulatory subunits of protein kinase A. Okay, so if you want the catalytic subunits to actually be active, what you need to do is release them from the claws of these regulatory subunits of protein kinase A. Now, how do you do that? With cyclic AMP. So cyclic AMP can bind into these binding sites here, and it causes a change in conformation in the regulatory subunits of protein kinase A, which I'll show here. Okay, so I'm going to show this dramatic change in conformation as them going from being in an L shape to being straight, like so. Okay, so the regulatory subunits of protein kinase A have been forced to undergo this vast conformational change by the binding of the cyclic AMP in these cyclic AMP binding sites. So two cyclic AMP molecules bind to each of the regulatory subunits. So these are the cyclic AMP molecules just shown as little blobs in these binding sites. Okay, that then releases the catalytic subunits from the regulatory subunits, and note the regulatory subunits still remain in their regulatory subunit dimer here, okay, but they release the catalytic subunits, and each of these two catalytic subunits can then go on to phosphorylate serine and threonine residues within proteins. Okay, so these are now serine, threonine, kinase enzymes. Okay, right. So the initial mechanisms that we are going to look at for the sensitization of nociceptors, including the mechanical and thermal nociceptors and the polymodal nociceptors, is going to involve these catalytic subunits of protein kinase A phosphorylating certain proteins. Okay, so the first target that I'm going to mention is the alpha subunit of voltage-gated sodium channels NAV 1.8. Okay, so remembering right back to the days when we were talking about voltage-gated sodium channels, and I'm just trying to find the picture. Here it is. Okay, when we were talking about voltage-gated sodium channels in primary nociceptive afferent neurons, I said that the three main ones which were expressed were NAV 1.7, NAV 1.8, and NAV 1.9. Protein kinase A seems to be able to phosphorylate the NAV 1.8 alpha subunit of voltage-gated sodium channels. Now, what is the effect of this phosphorylation? The effect is that it's going to cause this alpha subunit to open at lower threshold potentials. Okay, now what does that mean? That means that the depolarization that you have to build up in from the receptor potentials to actually activate an action potential is now going to be reduced. Okay, so let me get another picture back again. Not this one. What do I want? Not that picture either. Or do I want this? Where is that picture that I actually want? Okay, here, this is the picture I want, sorry, yes. Okay, so remember, in these free nerve endings of the nociceptors, what has to firstly happen is you have to get these receptor potentials caused by the excitatory currents coming in through these receptors that have been activated by the noxious stimuli. Okay, the receptor potentials then have to produce sufficient depolarization in this region that is capable of actually firing an action potential, and you have to get it up to threshold potential. If we have now lowered that threshold potential by phosphorylating the NAV 1.8, then it follows that less receptor potentials are going to be needed in the nerve-free ending to cause the firing of an action potential here, i.e. these neurons will now fire action potentials in response to smaller stimulations. Okay, that's allodynia. Stimuli that previously wouldn't have produced an action potential in these nociceptors is now producing an action potential. In addition, what it will cause is hyperalgesia, because stimuli that were big enough to produce enough receptor potentials that you get enough depolarization here to get up to threshold potential will now, under this new schema, produce more frequent action potentials there, and therefore you'll get hyperalgesia as well. So that's one of the mechanisms by which activating these EP4 receptors and then activating this GS cascade can lead to hyperalgesia and allodynia, i.e. sensitization of these nociceptors. Okay. Now, we have another one of these sensitization mechanisms which involves TRPV1 to go through. Okay, and then what we'll do is turn our attention onto how you can get these rhythmic action potentials being produced in polymodal nociceptors through this cascade, but we'll do that in the next video.